In this video, we're going to cover drug metabolism. We're going to look at the processes involved in drug metabolism, the reactions, and the key enzymes. We'll also explore the primary mechanisms that alter drug metabolism. But before we define what metabolism is and why the body metabolizes drugs, let's first look at this overview diagram here. If you've seen the introduction to Pharmacokinetics lecture, we went through an overview of the four important processes, A, D, and E. Drug absorption is the process by which a drug enters the bloodstream from its site of administration. Once a drug enters the bloodstream, it's distributed throughout the body to its target tissues. And now we're on to drug metabolism. So let's subtract complexity and break down what metabolism is and where it occurs. Okay. We define metabolism as the biotransformation of a drug in the body. By biotransformation, we mean a chemical reaction that the body performs on the drug, okay? The next question then is, why does the body metabolize drugs? For drugs to pass through membranes and barriers in the body, they must be lipid soluble, allowing for absorption and distribution throughout the body. We covered this in previous lectures. Now, the goal of metabolism is to make the drugs more water soluble so that it is easier to excrete them through the kidneys and urine. And what transforms drugs to make it more water soluble? So the liver houses the metabolic enzymes responsible for this transformation. And what these enzymes do is make the drugs more polar so that they are easier to excrete by the kidneys. Okay? The next question is, where does metabolism take place? It mainly takes place in the liver, but it can also occur in almost every tissue in the body. So including the muscle, fat, and gut wall. Okay? Let's draw this out. Let's say we have a lipophilic drug. This drug will reach the kidneys and be essentially filtered here. However, because it's lipophilic, it will be able to re-enter the body by crossing the walls of the tubules of the kidney. So it will be reabsorbed and it won't be excreted. Okay? This is what will happen to the drug regarding excretion once it's absorbed if it's not metabolized. Okay? So let's see what happens if it is metabolized. Let's take the same lipophilic drug. The lipophilic drug undergoes biotransformation into a hydrophilic metabolite through the action of metabolizing enzymes. Okay, so once it enters the kidney, it primarily remains in urine due to its inability to cross the kidney tubule walls and undergo reabsorption. Okay, so this shows how the body metabolizes drugs to become more hydrophilic so that they can be better excreted by the kidneys. Beautiful. The thing is, the extent of metabolism is not the same for everyone. Metabolism can vary with age, pregnancy, and disease conditions. The extent of metabolism varies from person to person, and this is because different individuals will secrete different levels of metabolizing enzymes. All right? Now, the body undergoes two primary metabolic reactions, known as phase 1 and phase 2 reactions. Phase 1 reactions generally prepare for phase 2 reactions, and what I mean by that is, let's say we have a drug X. This drug is lipophilic, and remember the goal is to make it more hydrophilic, more polar, and water soluble. The way the body is going to do this is perform a phase 1 reaction on this drug here. It's going to attach a functional group, generally a hydroxide, okay, an OH. Now that there is this hydroxide OH group, the body can perform a conjugation attachment, which means it will attach a large hydrophilic group to the drug molecule. That is the group A here. And as a result, the body will become more hydrophilic, okay, more polar, more water soluble, and it can be excreted by the body. Now here's the thing. The kidneys can excrete phase 1 metabolites if they are sufficiently polar. So if the drug is already polar and water soluble, it can be excreted. However, when phase 1 metabolites are still too lipophilic for excretion, we transition to phase 2 metabolism. And another important thing to know is that not all drugs are metabolized by the body. Why is that? Well, as we said, the point of metabolism is to make a lipophilic drug more hydrophilic. Again, we're going to keep saying this over and over again, more polar and water soluble. If a drug is already hydrophilic, there's no point in having the body metabolize it, right? Because it's already hydrophilic. So some drugs are excreted unchanged in urine, and that means they're not metabolized, okay? So here you've got a few examples of drugs and the extent of metabolism on these drugs. So if it says that a drug is excreted 100% unchanged, that means there is no metabolism performed on that drug. We'll break this down further in a minute because I want to go through the three main kinds of metabolites, but just keep in mind that if a drug is already hydrophilic, it's water soluble enough, it's polar enough, that the drug is excreted 100% unchanged, okay? It means that there is no metabolism performed on that drug. Okay, the products of metabolism are called metabolites. When a drug is metabolized, we get metabolites. First, we have inactive metabolites. So once it's metabolized, the drug loses its active properties. Second are active metabolites. And here we introduce the concept of prodrugs. This is where the drug becomes active after the body processes it. A great example of this is codeine, which changes into morphine in the body and becomes active. Another example is cortisone. It's inactive, but it's transformed by the body into hydrocortisone, okay? So to recall the concept of a prodrug, think of codeine being converted into active morphine by liver enzymes. And then the third type are toxic metabolites. A typical example is paracetamol. Some of it changes into a toxic form due to liver enzymes. This toxic metabolite is why excessive paracetamol can lead to liver failure. If an individual were to take a high dose of paracetamol, then there will be a high dose of this toxic metabolite, which could cause liver failure, okay? So remember our purpose here. Metabolism's goal is to make the drug more water soluble, making these metabolites easier to excrete, okay? But the thing is, there are some drugs that are already water soluble enough that the body doesn't metabolize them. There's no point, okay? In these situations, the drug is excreted in the urine without changing. The drug is already hydrophilic, so there's no point for the body to metabolize it. We're just gonna keep saying that, all right? Now that we've talked about the different kinds of metabolites, let's break down the two main types of metabolic reactions further, starting with phase 1. But remember our goal here. Remember the goal for metabolism. In the first step, lipophilic drugs are changed into more polar molecules by adding or unmasking a polar functional group, such as a hydroxyl group. Okay? Again, the purpose of these reactions is to make the drug more water soluble and also ready to react with the phase 2 metabolic reactions to make the drug more readily excretable by the body. Then this is where the cytochrome P450 enzymes come in, often abbreviated as CYP enzymes. They catalyze these reactions, phase 1 reactions. Scientists gave them these names because they are found within cells, so cyto, and also because when they're excited and they light at 450 nanometer, they're going to give a color to this solution. That's where the name CYP450 comes from. And CYP enzymes, CYP is a superfamily of heme-containing enzymes. They are proteins like all enzymes and are located in most cells, but primarily in the liver and GI tract. Let's talk a little bit about CYP enzymes. So in this family of enzymes, there are different subtypes that are going to target different drugs because many different genes encode multiple enzymes, okay? There are many different P450 isoforms. Each CYP enzyme is designated by a unique family, subfamily, and individual gene symbol. So for example, CYP3A4 belongs to the CYP3 family, the A subfamily, and the second number tells us the specific isozyme. All right, so this nomenclature system helps in categorizing and identifying CYP enzymes based on their structural and functional similarities, okay? And there are many subtypes of CYP enzymes. However, there are actually two subtypes that metabolizes 50% of drugs on the market. These two enzymes are the subtype CYP3A4, which metabolizes around 30% of the drugs on the market, and CYP2D6, which is involved in metabolism of codeine, which metabolizes around 20% of the drugs on the market. An important thing to mention about CYP enzymes is something we call genetic polymorphism. Okay, what does this mean? Why are we talking about this? Polymorphism means that there will be some variation or differences in the levels of these enzymes depending on the people. So there is inter-individual variation in drug effects because not everybody will have the same levels of CYP enzymes. We're all unique, right? One example is CYP2D6, which some people will metabolize more than others since different subgroups within the population have varying capacities for carrying out specific metabolic reactions. These inter-individual variations are caused by genetics and mutations in the genes that code for enzyme expression, meaning that some people will secrete more enzymes than others. So for instance, some people get no benefit from the opioid painkiller codeine, okay, because they lack the CYP2D6 enzyme, which is necessary for the drug's activation. So the genetic differences of CYP enzymes has significant clinical implications for drug therapy. Pretty cool, beautiful. Okay, that's a bit of background on CYP enzymes. Now, there are three main types of reactions involved in phase 1 metabolisms. We have oxidation, reduction, and hydrolysis. The goal of these reactions is to make a drug more polar and water soluble by adding an hydroxyl group or an oxygen. Okay, that's phase 1 metabolisms with the reactions being catalyzed by CYP450 enzymes. The kidneys can then excrete phase 1 metabolites if they're sufficiently polar and water soluble. However, phase 2 metabolism occurs when phase 1 metabolites remain too lipophilic for excretion. So recall our overview diagram looking at phase 1 and phase 2 metabolism. And that leads us to phase 2 metabolism. Phase 2 metabolism typically follows phase 1, although some drugs may undergo phase 2 conjugation directly, okay? So let's say we have a drug here that is active. The cytochrome P450 system renders it inactive by introducing a poly group, but it's still not polar and water soluble enough. So then we move on to phase 2. And in phase 2, our goal here, again, is to make the drug more polar and water soluble so that it can be easily excreted by the kidney or bile. This phase involves conjugation reactions where endogenous molecules are added to the group or its metabolites to increase their water solubility and facilitate their excretion from the body. And it's catalyzed by transferase enzymes. These enzymes facilitate the transfer, keyword being transfer, transferase, transfer, of specific functional groups to the drug molecule or its metabolites. Let's look at the main types of conjugation reactions. First, we have glucuronide conjugation. We're attaching glucuronic acid or glucuronate to the substrate molecule. Now the thing is, you can actually figure out which type of reaction it is by looking at the name of the enzyme. So for this reaction, the enzyme is glucuronyl transferase, which will transfer glucuronide to the drug. Next, we have the glutathione S transferase. This will transfer glutathione to the drug. Next, we have the enzyme sulfotransferase, which will transfer sulfate to the drug. This process is known as sulfation. And we also have the enzyme acetyltransferase, which will transfer an acetyl to the drug. This is also known as acetylation. So each type of conjugation will modify the chemical structure of the substrate, increasing its water solubility so it can be eliminated from the body via bile or urine. Okay? And again, you can figure out which type of reaction it is by looking at the name of the enzyme. Let's look at one example. Morphine is a typical example of glucuronidation. The process transforms morphine into morphine glucuronide, okay? Now, as you can see here, the name kind of looks like glucose, okay? And that's because glucuronide is a sugar moiety. So for a sugar that is very water soluble, attaching a glucuronide to morphine makes the morphine more water soluble. That's phase 2 reactions. Remember, our goal is to make a drug more polar and water soluble. Now what we're going to do is look at factors that influence this, that influence drug metabolism. All right? Earlier, we touched on genetic polymorphisms. Let's come back to it. So variations in DNA sequence among individuals that can influence drug metabolism. These polymorphisms may change the expression, activity, or function of enzymes that break down drugs, which can cause drug responses to vary from person to person. For example, genetic variants in CYP enzymes can lead to altered enzyme activity, affecting the rate and extent of drug metabolism. Remember, CYP enzymes catalyze phase 1 reactions. If we look at CYP2D6, where polymorphism is very important, the enzyme's level of production varies greatly between individuals. So some people are going to be ultra rapid metabolizers because they produce a lot of CYP2D6, and other people might be slow metabolizers because they don't produce a lot of CYP2D6, which makes sense, right? So let's look at drug interactions affecting metabolisms, especially through two mechanisms, enzyme induction and enzyme inhibition. We're also interested in the CYP enzymes, so we're going to look at inducers and inhibitors of these enzymes. Starting with enzyme induction. What are enzyme inducers? These are drugs or substances that stimulate the body to produce more enzymes. Now, the problem with increasing the number of enzymes is that it leads to increased metabolism, which can potentially increase the drug's toxicity, which makes a lot of sense. If you have a lot more of these enzymes, you're going to break down a lot of drug molecules, okay? So looking at my exceptional drawings here, the concentration of the active drug will decrease as we increase the P450 enzymes, thanks to enzyme inducers, which will increase the concentration of the inactive drug, okay? So what we're gonna do is we're gonna increase drug metabolism. So for example, if we were to enhance the metabolism of paracetamol, this will lead to the formation of more toxic metabolites, which in turn increases its toxicity. Another consequence of inducing drug metabolism, which can result in either low or no drug activity, is that the drug may not function at all due to excessive metabolism in the body. So inducers can be drugs, okay? It can also be influenced by various physiological factors such as age, gender, pregnancy, physiological conditions like liver or kidney disease, okay? There are also environmental factors such as diet, smoking, and exposure to pollutants. Dietary components and herbal supplements may contain compounds that interact with drug metabolizing enzymes. For example, smoking tobacco can induce certain CYP enzymes, leading to increased metabolism and decreased efficacy of certain drugs. This means that a drug such as theophylline, which treats the symptoms of asthma and other lung diseases, may not remain in the body for as long, okay, as smokers will metabolize it more quickly. As a result, this drug won't last as long and it might not even work for smokers. So if we add an enzyme inducer, that means that the body will produce more enzymes. The drug will be more metabolized, which means that the drug concentration may go down. Okay, let's look at an example. What happens if a patient taking warfarin is also taking the complementary medicine St. John's wort? The complementary medicine is an enzyme inducer of both CYP3A4 and CYP2C19. That means that taking this complementary medicine will cause the body to produce more metabolic enzymes, especially these two CYP enzymes right here. In the case of warfarin, that means it will be more metabolized or metabolized faster because we have more enzymes to metabolize the drug. So there is a risk of not reaching the therapeutic blood concentration or blood level of that drug. What does this mean for the patient if they were taking warfarin and St. John's wort? That means there might not be any therapeutic effect of warfarin. So that's a big risk for that patient, okay? So to summarize, in the case of enzyme inducers, the body produces more enzymes. So the risk is that the drug is rapidly metabolized because we have so much of these enzymes, and that the plasma concentration decreases. When the plasma concentration drops, the drug loses its activity. Okay, that's enzyme inducers and how we can affect CYP enzymes. Let's look at enzyme inhibitors now. Okay, enzyme inhibitors act in the opposite direction. What they do is they block or slow down enzyme action on drugs or molecules. They typically bind and inhibit the enzyme's activity. Okay, so we're working in the opposite direction now. They're blocking the activity of the enzyme. The problem with enzyme inhibition is that the drug will have a longer half-life, making it last longer in the body than expected. It will also prevent the metabolization and activity of a prodrug, like codeine, which requires metabolization for activation. And another problem is that because the drug is not metabolized, it can accumulate in the body and can eventually reach toxic levels. Okay, so the action of the enzyme will be blocked by the inhibitor, which means there will be less metabolism and the drug concentration may go up. Okay, pretty straightforward. If there's no enzyme, if we're blocking the activity of the enzyme, then the drug cannot be broken down. So enzyme inhibitors can be drugs, they can be pesticide additives, substance abuse, so alcohol or industrial solvents. There's also an influence of age, pregnancy, disease conditions. So there are several factors that can influence metabolism and the levels of enzymes that are produced by the body. Let's look at one example. Lipitor and grapefruit juice don't work together, okay? Lipitor is a statin, it's an anti-cholesterol drug, and if you look at the label of that drug, it's written to avoid eating grapefruit or drinking grapefruit juice, okay, while you're being treated with this medicine. Now, why is this? Grapefruit is an inhibitor of the CYP3A4 enzyme, which means that in the presence of grapefruit juice, this drug will be less metabolized or slower metabolized, which means it will last longer. It also means that there's going to be a risk of reaching toxic concentrations and seeing more side effects, because remember that this medication will be taken every day. So in the presence of an enzyme inhibitor, that drug, okay, so Lipitor, may start building up in the body because it's less metabolized. All right, so that's the risk of taking this drug with an enzyme inhibitor. What do enzyme inhibitors do again? They will block metabolic enzymes. Since they won't function as well, there's a risk that that drug won't be metabolized effectively. The main risk is that the drug's plasma concentration may increase and potentially reach toxic levels, which could lead to side effects or, in the worst cases, lethal effects. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video, or you can view the entire playlist.